I'm really excited to be here talking to you guys today and also just be part of the CAIRIBU program. Like Dr. Palasko mentioned, my background was not in urology at all, and I started to be interested in this project after working in Dr. Kim Kyle-Stietz's lab. I started some gene by environment interactions of autism work there in my postdoc, and then because her lab has such a urology focus, started to be interested in how voiding could be different in people with autism. So thanks for having me. I've really liked being a part of the CAIRIBU community, and I've just found everybody to be so welcome and supportive. And I've really gotten a lot from all the various events. So with that, I'll get started with my talk. So peeing is complicated, to go or not to go. This is a question that we all probably, think of a lot in our research. So here's kind of just a picture of how this whole process takes place in our body. So it's very complicated. Involvement between the brain, which is the pontine micturition center specifically, integrates signals to initiate voiding, and the cortex can override these signals. And this is called retention. And then there's an afferent pathway that senses bladder fullness. Sorry, just trying to minimize. Okay, that senses bladder fullness. And then there's also this efferent pathway that helps with retention, or holding behavior. So this maintains tension in the sphincters, promotes bladder relaxation. And then during voiding, it has to be this coordinated effort to contract the bladder and relax the external sphincter. So there's really a lot going on for a successful retention behavior, being able to hold your bladder when you don't want to go, and being able to actually void completely when you do want to go. So errors in the control of this process lead to LUTS. So LUTS is made up of a lot of different things, can be increased frequency, residual urine, going at night, incontinence, or retention behavior. And LUTS can affect everybody. So this is really common in aging populations, also common as a result of childbearing. Children can have LUTs, such as bedwetting, and then children and adults with neurodevelopmental disorders frequently have LUTs. So actually 77% of persons with ASD report having LUTs. So when I was thinking about this idea, this is kind of what got me going, because there was this big study that looked at how often people with neurodevelopmental disorders have LUTs. And it's 77%, which is obviously a vast majority. So this is not an uncommon problem in this population. So I'm just going to talk a little bit about autism spectrum disorder, so we have some background for it and why it might contribute to LUTs. So this has a prevalence of 1 to 2% worldwide, and it's characterized by difficulties with socialization, repetitive behaviors or tics, restricted interests, tactile hypersensitivity, and also, in this group, we'd like to add urinary dysfunction to this list as well. It has a really complex etiology, which just means that there's a lot of factors that go into whether or not someone will develop autism spectrum disorder, which I'll come back to in a little bit. In terms of pathophysiology of autism, in general, it's characterized by altered neuronal connectivity, underconnectivity in childhood, and overconnectivity in adulthood. So on this diagram here, in blue, we have the connectivity of an individual with autism spectrum disorder, and then a typically developing person in this peach color here. And in childhood, there seems to be too many neuronal connections, and then in adulthood, there seems to be too few. This is a little bit dependent on the age of the person that you're measuring in, and also the brain region. And this observation is kind of further limited by, we have a lot of fMRI studies to characterize the functional connectivity of the brains of people with autism, but obviously we can't look inside their brains. So it's, all these observations are limited by that, and there's a lot of room for new research ideas in terms of characterizing these differences in connectivity. One thing that people have posited lately is that adolescence, this oval here, could be a vulnerable time of neuronal plasticity, which just means obviously this change has to happen at some point. So it seems like like late childhood, early adolescence, there's this pruning that should happen that might not happen, or maybe they don't have a process that should happen. So um, it's very complicated and a lot of room to grow. But just to talk a little bit about the molecular basis of neuronal connectivity, um, and I realize that I'm giving a little bit more background, maybe just because this is, primarily a urology group. So I just kind of wanted to give a little bit of a background about things that we've probably all heard about before, but just like to contextualize it for this talk. But neurons are connected in a network and by their dendrites. So this is a picture of a couple neurons that are connected together. And the dendrites up here are the little tiny branches, and they're kind of like the satellite signal that actually receives connections from other neurons. So this is a very zoomed in version of what we call a dendritic spine right here. And so this is one functional unit of connectivity in the brain. So this dendritic spine connection here. And dendritic connectivity is actually a very dynamic process. So what the machinery does here, all these proteins that I have listed, they connect with the NMDA and AMPA receptors, which receive glutamate excitatory signaling, and then they actually couple to the actin skeleton to form and eliminate spines. And then this results in alterations in the connectivity. This allows the brain to make and lose connections in response to stimulation or lack of stimulation. Um, so this process is disrupted in autism spectrum disorder, and this is likely what underlies the fact that we have this observation that brains are overconnected at some points and underconnected at other points in autism spectrum disorder. Okay, so the complex etiology of autism. So there are about 1,000 genes that are risk factors for autism spectrum disorder. And gene by environment interactions have also been posited to contribute to who gets autism spectrum disorder or not. So that's a question I'm really interested in, because everyone has a genetic makeup, but how does what you encounter in your daily life, or perhaps when your mother was, pregnant, or the environment that you have at home, how does that impact the likelihood or the severity of the autism spectrum disorder that you might experience? So many of these genetic risk factors are in genes controlling synaptic connectivity. Getting back to my picture here. So what I'd like to talk to you about today is the Shank family of proteins, which accounts for 1 to 2% of these mutations. So the Shank protein I have with a star right here. And this protein is part of the machinery that helps couple the excitatory signaling to the actin skeleton. And there, again, 1 to 2% of people with autism have a mutation in this pathway. And then there are also other proteins along this pathway, like PSD95, neuroglians and neurexins, that also have genetic risk factors for autism in them. So this pathway kind of represents a clustering pathway of genetic risk factors for autism. Okay, so that brings us to the Shank3B mouse model of autism, which the first question I usually get when I'm presenting at one of the CAIRIBU conferences, which is such an interesting question, just for perspective, is like, well, how do you model autism in a mouse? And how this is done is that they took one of the mutations that was found in the Shank proteins in this pathway, and then they mutated this in a mouse. And so this is a knockout model, and they replaced this big chunk of the protein and so this results in a non-functional Shank3 protein. So that is the model of this is one of the models of autism that people use to look at autism behavior in a mouse. So these mice have reduced time they spend socializing with other mice. So normal mice will spend time socializing and being next to other mice, but that process is disrupted in this mouse model. They also have increased grooming behavior. Um, and then they also exhibit increased dendritic complexities because obviously we can look inside a mouse brain. So these mouse models also have increased dendritic complexity. So we have a lot of the characteristics that we think um, presents as autism in people in this mouse model. So that is kind of how we look at autism in this mouse model. So for my first experimental goal when I started the lab was to establish the Shank 3 ASD model so that I could look at, answer all these questions that I was interested in answering. First, we have to establish that this actually, we can actually measure some of these autism phenotypes. So this is a pretty easy experimental timeline. Basically, the mating pairs are set up heterozygous to heterozygous, and the offspring is born. And then at 6 weeks of age, we look at the autism behavior and voiding physiology all within the six week. So the first task that I want to talk to you about today is called the odor discrimination task. So I put the mouse in a cage, and then I present them with Q-tips that are dipped in certain scents. So the first one is water, and then vanilla, and then same-sex urine and opposite-sex urine. And what we would expect to see for this is that we are measuring communication with the mice because social odors are how they communicate. So wild-type mice should have a strong preference for urine scent over control. Shank 3 heterozygous mice should have a moderate preference, and then Shank 3 homozygous mice should have a low preference. And when I do this, this is what we see, which is really exciting. So response to social odors is diminished in both male and female Shank 3 heterozygous plus minus and homozygous minus minus mice. So in female mice here, we have water on the far left for purple, which is wild type, Shank 3 heterozygous, which is blue, and then Shank 3 homozygous, which is green. Not very much response for either our negative control, water, or positive control, vanilla. And then for same-sex urine, we have a large response from the wild-type mice, and this response is diminished compared across the genotypes. Why did it? Oh, we went back. Okay, sorry. So then with the male mice, we also see a very similar trend. So really large response to the same sex and opposite sex urine in the wild type mice, and then a diminished response in both the heterozygous and homozygous mice. Although the heterozygous aren't significant, they're really close to being so. So getting back just to summarize, this is what we expected to see, moderate preference, low preference for urine over control. So this is recapitulating what has already been established here. Next task that I did was a 3-chamber social approach, and basically the mouse is put into a chamber that has 3 different sections. There's a novel mouse on one side and a novel object on the other side under a cup, and the mouse is left to freely explore the arena. And this is a measure of social behavior because wild-type mice will spend more time in the chamber with the mouse under the cup than any of the other chambers. So we would expect a wild-type mouse to have a strong preference for novel mouse over novel object, and then heterozygous mice to have moderate, and then again, homozygous to have low. So here what we have is female mice on the left, male mice on the right, and then in purple we have wild type, heterozygous in blue, and then homozygous in teal. And then on the far left we have the amount of time that they spent in the center compartment, and then the middle bar for each of these graphs is novel mouse, and then the right bar for each of these subsets is novel object. And so what we would expect to see is that the novel mouse bar is much higher than the novel object bar. So for some reason, the wild type mice weren't showing this phenotype. However, the heterozygous mice were really strongly showing this phenotype. And then the homozygous female mice were not showing this phenotype, which is what we would expect. And then for male mice, both the wild types and the heterozygous spend significantly more time with the novel mouse. And then this was not significant for the homozygous mice, although we do need to, I need to expand the N values here. However, overall, we can say that this is about what we expected, with the exception that the wild type female mice didn't exhibit a preference. Okay, the third task I'm going to tell you about quickly is repetitive grooming. So here I just have a little video of a mouse grooming itself. Now, all mice do this, but mice with certain autism spectrum disorder mutations tend to do this more, and they actually overgroom themselves and give themselves like dermatitis and bald spots. Okay. So what we'd expect here, this is demonstrating tics and repetitious behaviors. So wild type mice would spend some low amount of time grooming, moderate amount, and then high amount for the Shank3 mice. So the females here, the wild type is in purple here, so really no difference with the heterozygous mice. And then although the homozygous mice here is not significant, it was trending to be significant. And then with the male mice, there is an increase with both the heterozygous and homozygous mice, although again, the homozygous is not significant due to the N values here. So overall, I would say this is also, for mouse behavior, recapitulating what we expect to see. So conclusions for this section is that I think I can say that the autism phenotype assessment was recapitulated, and the females overall are a little bit less reliable than the males, which is really interesting because female autism is less common than male autism. Males are about 4 times more likely to get autism than females, and female autism is definitely something that is less researched, and a lot of the work that was done in this original mouse model is actually all in male mice. So something kind of interesting there. I would also say the heterozygous Shank3 mice demonstrated a moderate phenotype. So this is really good for my gene by environment interaction models because I am interested in a genotype that perhaps wasn't severe to begin with, but then given some sort of environmental insult would be severe. So that is a project I'm working on in a different capacity. But overall, I was pretty happy with how this data turned out, and it's really exciting, as a postdoc to get my own project off the ground that I had thought of and everything. So, really excited about that data. So, goal 2 was to investigate LUT's lower urinary tract symptom phenotype in Shank3 ASD model. Before I had mentioned that I'm happy to take questions during if anyone would like to talk or ask a question. So I'm going to pause if there's any questions before I move on to the next section. Okay, I'll keep going. Okay, so goal 2 was investigate this LUT phenotype. And to do that, we're doing void spot assay. And basically the mouse is set into an empty cage with chromatography paper. The mouse is left to freely urinate, and then we quantify the urine spots using UV imaging and then void wizard analysis. So from that, we can get like the number of spots, total area of spots, and the distribution of spots. So this allows us to assess voiding dysfunction while the mice are awake and kind of doing their normal behavior. And so for wild type mice, we would expect to see urination in the corners, some large and some small voids. So what we maybe would expect for the Shank model, although we, really didn't have a concrete guess about that because no one has done it before, was that we might see a difference in spot size distribution, or more in the center and less in the corner, because voiding in the corners is a behavior that is common in mice. So, perhaps this was disrupted in the autism model of mice, and then again, just more pronounced for the heterozygous. So what we do see is that female mice and male mice also display less small spots in the void spot assay. So on the y-axis, we have total spot number, and then on the x-axis, we have the bins for the spot size. So on the far left is the smallest spot size category. And here we see that the females exhibit less small spots with the genotype manipulations than their wild type counterparts. And this is also true for the heterozygous mice here as well, and then trending for the homozygous mice. Other parameters that we get from this assay are spot area. So none of this was significant when I ran the stats. However, we do need to expand the N values here a little bit. So spot area was slightly increased, which makes sense. Slightly increased here for males, but not females, not significantly. Percent urine in the center was not very much different. Which is kind of interesting. I might have expected it to be actually a little bit increased for the SHANK3 mutants. Percent urine in the corners, maybe a little bit down for the male mice, but again, not significant. And then the number of spots was less for the heterozygous mice. For females, it was almost significant, and it's trending for the other groups, which is mostly driven by the fact that there's less small spots. Okay, so for this summary, we have that there are less small spots in both the heterozygous and homozygous mice, with generally the homozygous being more pronounced. Not a real difference with center or corner voiding, but we do need to increase the N value here and see if it will be significant after that. Okay, so the next thing that I'm looking at is anesthetized cystometry, which is CMG, abbreviated as CMG. So this looks at voiding physiology while the mouse is under anesthesia. So what we do is we take the bladder out, and then we put a small tube into the bladder and then sew it shut so that the bladder is filled with saline. And then we can look at the bladder pressure on the computer as the mouse goes through rounds of having its bladder filled and then voiding it out. So this is what the readouts for this experiment look like. So this is a void, this spike and then drop down here. So void, I'm calling a void 0 on the left and then void 1 on the right for the analysis. So if we're quantifying void 1 here, then some of the parameters we're looking at is baseline pressure, which is the pressure that the mouse returns to after the previous void. Threshold pressure, which is the pressure right before the void is initiated. And then the maximum threshold, which is the top pressure that the mouse reaches here during voiding. And then intervoid interval is just the time in between, void 0 and void 1. So we would expect differences in voiding here if the voiding dysfunction in the autism model is not purely behavioral, which is a good way to get at the two ends of this problem, because there are, it's also been posited that, well, people with autism spectrum disorder just, have some sort of LUTs because of behavioral problems, not because there's anything different actually in their bodies that's causing them to have a more difficult time with these sorts of LUTs. So regular interval, so for wild type mice, we'd expect regular interval voiding with large voids and then minimal non-voiding contractions. Not exactly sure what we maybe expect for these before we started, but I was guessing maybe irregular intervals, increased pressure threshold, or decreased voiding perhaps. And then again, more pronounced in homozygous mice. So this is a preliminary result because they only have one mouse replicate per group, but preliminarily it kind of looks like the SHANK3 homozygous mice exhibit increased pressure thresholds and more non-voiding contractions. So the wild type on the top plus, heterozygous in the middle, and then on the bottom is the homozygous mice. And you can see they have this really big kind of like arm to their voids, and that is the increased threshold pressure right before they void. So it seems like their bladder has to get fuller perhaps before they can sense the voiding. So here's the quantification of these parameters here. Again, I haven't run any stats yet because it's just an N of 1, but maybe a little bit decreased for the intervoid interval with the SHANK3 mutation, trending towards increased number of non-voiding contractions for the SHANK3 homozygous mice. The non-voiding contractions are these little spiky parts, like on the arms of the void. And then the maximum voiding pressure didn't really seem to look very different here, but the normalized threshold pressure was trending towards being increased in just the homozygous mice. So I explained what we would expect to see here. Not a huge difference with the heterozygous mice, but there was a trend towards increased pressure threshold and more non-voiding contractions in the homozygous mice. So conclusions for voiding physiology here. The void spot assay shows a reduction in small spots in both heterozygous and homozygous mice. CMG shows a trend towards increased pressure thresholds and non-voiding contractions. So this may indicate that the afferent bladder sensing is less sensitive than it should be, and the combination of these two observations indicates a less sensitive / retention phenotype. So that means that perhaps these mice are not able to sense that their bladder is full, so they're holding on to urine longer than is normal, is kind of the thought here. Okay, now I have some kind of preliminary data to show you about things that I've started working on specifically for the K12, which I'm really excited about. So this goal is to test the hypothesis that differences in neuronal excitability contribute to ASD-associated LUTs. And so again, to get back to this picture, it really could arise from any of these things, and this is a very complicated system, but since there's not very many people looking at, I call it autism-related LUTs, we decided to look at a couple different areas and see if there was differences here. So for my project, I'm looking at the pelvic nerve excitability, dorsal root ganglion, and then also the pontine micturition center, but spoiler, that's going to be a future direction because I haven't gotten around to that. But this is really because I came from a neuro background and I've always been really interested in, how the brain controls the rest of your body. So I was really interested if the neuronal excitability of these specific areas contributed to voiding dysfunction in autism. So also super excited about these experiments. We were actually able to record from the pelvic nerve. So there's actually a little electrode that we put underneath the nerve. And then the animal is hooked up to the same pressure transducer, and we can see the voids happening. And then you can also see that the nerve is spiking during this time, like right before, this is pelvic nerve, right before the animal voids. So special shout out to Dr. Nick Burgraff and his whole lab at UW-Madison and CBS for helping us with this. I've learned so much about nerve recording, and also shout out to Kim because she is amazing at finding the pelvic nerve, which is really difficult. So I feel super fortunate to have both Nick and Kim to learn from and to help me with these experiments because they're very technically difficult, and it's just great to have so many resources available to me. So future directions for this is analyzing the time, maybe the time constant between spike and void and other spiking parameters. So I'd be kind of interested to see if there's like a difference in how much time there is in between the neuronal spiking and then the void. Sorry if you can hear my dogs in the background. I think my husband just got home. Okay, so also I'm looking at dorsal root ganglion neurons cultured from SHANK3 homozygous mice. And so what I do here is I actually take out the dorsal root ganglion and then I culture it for 7 days. And then I stain it with something called beta tubulin, which actually shows the total neurite outgrowth in cultured dorsal root ganglion cells. So the green represents just all neurite outgrowth, and I've only done this twice, so this is like a representative images, but both times the homozygous mice looked like they put out more neurite outgrowth in those 7 days. So future directions for this are that, I'd like to complete at least an N of 3 for each genotype, and then analyze normal neurite staining to cell culture. Okay, so goal 4 is to, um, sorry. Okay, goal 4 is to test the hypothesis that zinc supplementation improves this LUTS phenotype. So, um, why zinc, you might ask? So, SHANK3 actually requires zinc to form tertiary sheet structures in the postsynaptic density. And what I didn't tell you earlier is that the region that's actually um, modified in the SHANK3 model is the zinc binding domain. So with zinc, the SHANK3 proteins are able to actually form a sheet in the postsynaptic density. And then without zinc, they're not able to form this sheet. So zinc deficiency may contribute to phenotypic severity through gene by environment interactions. And the hypothesis would be that low zinc would result in a worsened phenotype, and then high zinc would improve phenotype severity. We also have evidence that zinc supplementation improves ASD-related phenotypes in these SHANK3 mice. But of course, our question is, well, what about voiding? Would it help that? So right now I'm working on experiments where the mice are on these modified zinc diets from the time that the mating pairs are set up all the way through the time of testing. And I have a low zinc, normal zinc, and a high zinc diet here. And then at 6 weeks of age, again, we do the VSA, pelvic nerve, and tandem CMG, and then also the DRG cultures. So this graph actually that I've showed you before, this shows the normal zinc conditions. And then I also have low zinc, which is in purple, and then high zinc, which is in pink. And then on the far left is the wild type mice, the plus plus, heterozygous in the middle, and then homozygous on the right. So for intervoid interval here, maybe a little bit of increase with the low zinc, like a longer duration in between the intervals. Again, I know this is a really, really low end value, so not reading into it too much. But the number of non-voiding contractions for all 3 of the zinc groups in the homozygous mice were all a lot higher than any of the wild type or heterozygous groups. And then also maybe the high zinc is kind of increasing the number of non-voiding contractions. Zinc is also really important in a lot of neuronal excitability contexts, so it perhaps wouldn't be surprising if too much zinc makes your circuits a little bit hyperexcitable. So normalized threshold, again, all 3 of the homozygous mice here exhibited some sort of increased threshold as compared to the other genotypes. And then the maximum voiding pressure wasn't super different with the genotype or the zinc. Okay, so conclusions and future directions. Conclusions recapitulated the ASD mouse model, got that up and running here. I would say that the heterozygous mice provide intermediate model, and the female behavior is a little less reproducible than the male behavior. Which a lot of people, ask me, well, could that be due to their estrus cycle? And it absolutely could be. I did not track their estrus cycle, but that definitely could be. So the mice with ASD risk factor mutations display altered voiding that may indicate a retention phenotype. That's what our preliminary data is saying. And we're investigating if zinc could help rescue that at all. So for future directions, obviously just kind of expanding all the values for the zinc experiments that I'm doing, begin calcium recording experiments for the PMC, and then in addition to culturing DRG excitability, I'm also going to do a micro electrode array experiment where I actually culture the DRGs onto a circuit board that will let me test how excitable the DRGs are. So with that, I would just like to thank Kim, Kyle Steets for being a great mentor and giving me such a great place to learn. We recently went to SOT together in San Diego, which was really nice. But just the entire lab, everyone is really helpful, and we really do group science in Kim's lab, which is great, especially when I have a hard stop for daycare pickup every day. I have definitely relied on the awesome grad students to help me finish up stuff after I have to leave. So really grateful to all of them for always pitching in and being such great coworkers. We recently went out for waffles as a group, and that was super fun. So it's, great to do good science, and then also have people that you enjoy as people as well. Funding sources, obviously, I'd like to thank the K12 for this amazing opportunity. Really just learning so much from you guys, and really grateful for all of the resources. And then Kim's R1 and R00. And then also I'd just like to thank the mice. I think that's important too, since they're kind of unwilling participants in our research, but we certainly learned so much from them, and it's a really important tool that we have.